Namaste students. I am JBN Dr. Rajesh Mohammed Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapeeth Women's University, Jaipur. Today I am going to discuss about the topic separation of serum from the blood samples. So our experiment is actually the separation of serum from the blood, but I will also explain the separation of the plasma from the blood. We talk about the separation of blood and the separation of serum and plasma, then we need to understand that what is serum and what is the plasma. In the blood, some part of the blood has plasma and some part of that may have serum. It depends on the processing of the blood sample when we are processing the blood, and on that basis we can isolate either serum or plasma. And if we process the blood immediately after the isolation, after collection, then we receive plasma. And if we do not process the blood immediately after collection and allow the blood to clot, then we get serum. So blood plasma is the liquid component of the blood in which the blood cells are suspended. It makes about 60% of the total blood volume. It is composed of mainly water and contains dissolved proteins, glucose, clotting factors, minerals, hormones, carbon dioxide, and plasma is the supernatant fluid obtained when the anticoagulant blood has been centrifuged. So the centrifugation is the main, very important thing in the collection of uh, plasma and the collection of the serum. The blood is mixed with an appropriate amount of anticoagulant like heparin, oxalate, or EDTA, and this preparation should be mixed immediately and thoroughly to avoid blood clotting. Blood serum is the blood plasma without fibrinogen or other clotting factors. So if we to add some clotting factor, then we receive plasma, and we process the blood immediately after the collection, then we receive plasma, and we process the blood group after half an hour or one after 1 hour, so that the blood will get uh, agglutinated, coagulated, and after that when we centrifuge the blood, then we isolate serum. So serum is the coagulated form of the blood by which we isolate the coagulated blood by centrifugation. So this is the idea how we get serum and how we get blood plasma. Now go to the procedure of the isolation of blood and isolation of the plasma. If we want to isolate the plasma, so we can go to plasma isolation procedure. First, we draw blood into the centrifuge tube containing approximately 1.8 gram 8 mg of the potassium EDTA. So potassium EDTA is an anticoagulant agent, and we must be sure that the full volume, we take the full volume to ensure that the correct blood to anticoagulant ratio, and then we invert the tubes. Okay, we invert the tubes 10 times carefully so that the blood and the anticoagulant mix together, and we store at room temperature until the centrifugation. Samples should undergo centrifugation immediately. If we process the if we process for the plasma, it must be centrifuged immediately, and this should be carried out for a minimum of 10 minutes and 1000 to 2000 RC, 13 RC at the room temperature. And it goes some three layer. After centrifugation, we receive three layers. The first layer is for the plasma, second layer is for the leukocytes, and the another layer is for the erythrocytes. So first layer has plasma, leukocyte, and the erythrocyte. Then we carefully aspirate the supernatant, the upper part. Okay, upper part. First layer, second layer, third layer. We carefully aspirate the upper part, which contains the plasma, and pull in the centrifuge too. We take care that we do not disturb the cell layer while transferring any kind of cells. We then inspect the plasma for its turbidity. Turbid samples may should be discarded, and the centrifuge and aspirant again should be removed. Then this is the process for the collection of the plasma. If we want to collect the serum, then what should be the procedure? First, we draw the whole blood into the centrifuge tubes containing no aggregate. Okay. First, we collect the blood sample. We allow the blood to coagulate. Okay. We allow the blood to coagulate. We do not process the blood immediately. Okay. And also, we are not adding any kind of anticoagulant. Okay. So first, we allow the blood to coagulate. Then we throw the blood sample into the centrifuge tube where no aggregate is added. Then we draw approximately 2.5 times of the volume which is needed for the use. Then incubate uh, the blood tubes in an upright position at the room temperature, that is 30 to 35, 45 degrees centigrade, and we allow them to coagulate. Then we centrifuge the blood at the 1000 to 2000 RCF volume. Then we have taken care about not to break to stop the centrifuge, because if we stop the centrifuge between the time, then it may disturb the process or for the isolation of the serum. After that, when the centrifugation has been completed, we carefully aspirate the supernatant. The supernatant will be the upper part, and we pull into a centrifuge to taking care not to disturb the another cells, so that if we disturb the another cell layers, we can isolate some another cells. So it must be taken care that isolate only plasma, not the cells. For isolation, we use a clean pipette and aspirate the blood. And we inspect the serum for the turbidity, and turbid samples should be centrifuged and aspirated again to remove the. If we found then there is a the sample is turbid, the serum is turbid, we again centrifuge it and also again aspirate the serum. Okay. What we observe, three layers are seen in the centrifuge tube containing the blood and anticoagulant serum. Serum was collected from the floating layer of the blood clotted sample. It will be a liquid yellowish uh, fluid. So we can mention over here results here that what is the color, what in which layer we have isolated. So the answer is simple. We isolated the serum from the upper layer. We isolated serum from the upper layer of the supernatant. So this is the experiment which I hope that I have explained, and what will be students will learn about. Students will learn that what is the purpose of isolation of the serum from the blood. How explain the complete experiment on the isolation of serum from the blood. Explain complete experiment on the isolation of the plasma from the blood. What is the difference between the isolation of the serum and isolation of the plasma from the blood? So this is the experiment, and uh, students will be able to understand about the process of the isolation of the blood sample, isolation of serum, isolation of the plasma, separation, and what precautions should be taken. They will also understand the ratio of the anticoagulant and blood ratio of the anticoagulant and the blood for the isolation of the plasma. So the plasma is not the part of this experiment, but I have added some part of the plasma in this experiment so that students can get aware about the isolation of plasma also. So I hope that the experiment is fully understood by the students. I will also explain again. So first, for the isolation of the plasma, we need to collect the blood sample, and after collecting the blood sample, we add we process the blood immediately, and we add anticoagulant inside the blood, inside the centrifuge tube, and we add the blood. This must be completed in a particular ratio, and then we invert the tubes 10 times so that the anticoagulant in the blood will be mixed together. And then after that, we centrifuge the blood immediately, and after centrifugation, we isolate some three layers, the first layer, second layer, and third layer. The first layer is useful for us because it contains plasma. Upper layer, supernatant, is the uh, layer where which contains plasma, and it must be collected. We aspirate the supernatant by the um, pipette, and this is our plasma. Now we go to the isolation of the serum. For the isolation of serum, what we do, we collect the blood sample, and we allow the blood samples to settle for 1 hour or half an hour. Then we transfer the blood samples into the centrifuge tube. And when we have transferred the blood samples into the centrifuge tube, we are not adding any kind of anticoagulant. No anticoagulant is added. And then we centrifuge. We allow the blood to centrifuge. Then the centrifuge has been completed. The blood is taken out. Tubes are taken out, and the upper layer is uh, aspirated. The aspiration is conducted by the some pipette. The supernatant is collected, and we take care that the some another layer should not be taken out. The care must be also be taken that if there is any turbidity, we do not take out the sample. If we find that it is turbid, then we again centrifuge the samples so that we can find proper serum and not any kind of cells. So this is all about the process. So my lecture is over here. Thank you very much.